Now for sampling, how do we even start? We talk about the case studies, the importance of bioavailability, but where do we begin? Now let's first start with a general procedures of sampling in general, but we can of course apply to soil sampling. Number one is to formulate the research questions. What do you want to ask? Why are we doing sampling? We don't start experiment without asking questions first to solve a little bit of cerebral entanglement in our mind, what we want to ask. If there's a problem they want to solve, we do it. But before we do, we think about it. We also have to obtain samples of soil from various locations after we have formulated the research questions. Now we collect the soil, we dry, and then we grind it in mortar and pestle. Make sure it's free of moisture. Right. Then we can digest it using aqua regia, concentrated acid. Okay. Right, to leach the metal. And finally, we need to use an analytical instrument. Normally, we use the ICP-AES, inductive decoupled plasma atomic emission spectroscopy, to test the level of metals. Then considerations when choosing a sampling locations. Critically is about safety. As researchers, when we go outfield study, always do risk assessment to ascertain whether the locations are safe. And then always keep in mind what are the questions you want to seek from your results here. What are the numbers and potentially what do they indicate? Representation and coverage. Would your locations be representative? Would the samples you choose from the location be representative of your answer, what you want to report, and your conclusion? So we do sampling outdoor, and in the case of soil, right, we need to visit the field, and you have seen the video by myself and a colleague. Now, let's say we have identified a field that's safe, right? On the campus or outside, you get permissions. And then there are three main kinds of sampling in general. One is point sampling. We just drop randomly, okay, throw a coin. Oh, these are the 3, 5, 6 points sites where we dig the soil and collect. Or we can just randomly put up some sticks, cut, okay, we have traverses, so it divides into the area and then we just pick a few areas, again randomly. Or we can do quadrants, right? Quadrants again, then we can do point sampling with it. Now, the design point sampling, right, is a bit more interesting. So if you are doing point sampling, there's 4 ways of doing that. Number one is just random, right? I look at the whole big area here, I just scattered my coins, okay? And then these are the places we do it. So it might be that certain parts are not covered because also random. Random doesn't mean it's average. Now for systematic, there's pros about it, which means that in every small localized area, there will be guaranteed a sample size, and because it's consistent, it's systematic such that it's regular, they put grid lines, right, the y-axis and x-axis, and then every particular corner is always the same spot, you see. So systematic and regulated, definitely we get a sample here, but it's not random. The next is the stratified random, which means you will have also grid lines, so x and y-axis, but you make sure that in every box that you have put a grid, it is random. So you go to every box and you start to throw a coin randomly. So you see now it's all scattered within the box, but you ensure that in every box there is a sample. And the last one is stratified random. Where you use it is that we know and aware that in the particular field, right, there's really sequestered zones, right, particularly sand, silt, and clay, that if you do random, then we are just fooling ourselves. No point bluffing us that, oh, okay, yeah, we have samples here. In the end, the result is very skewed because you will be only testing perhaps clay and silt. You miss out on sand, right? But now that with this information, you know that this is a impartial distribution of these kind of components in the soil, then we do stratified sampling, which means, okay, this is the soil part, okay, heavy on soil, come, let's do random over there, silt, the other side, and clay, okay. So it's highly important to know the theories of sampling on this slide in terms of the consideration. For the soil type identification now is a bit more practical, so you can do it outside if you have done risk assessment and it's approved. First, you take the soil, right, this from the nature, from the garden, you take a ball of soil and then you moisten it. How do you moisten it? You spit, okay, using your saliva. No, spit, not so courteous here. and then you roll into a ball, okay, like this. Try to roll it out as you roll to the ball. If yes, then you take a ribbon, you roll it, okay, about 1 mm thick. Okay, you try to roll it into the circle, and then use this table on the left here to identify the soil type. In general, to summarize, but my colleague and I have made another video for you to just go on and see. Make sure you don't have any meal before watching that video, okay? Because although it's au naturel, it's natural, but some of the observations are not very sightly to your appetite. Okay, so these are clearer representations. It's like a little flow chart here. Measure the size, yes, no, then is it sand, roll it, does it stick, right? Does it curl, does it break, does it form a circle? Can you form a U shape into ring? That's how you classify it, okay? Right, good. Now you get your hands dirty. You got your hands dirty just now, you wore a glove, right? But still it's dirty. Sample preparations. Now, we bring back to the lab here, what do you do here? So you collect the soil and then it dries in oven, okay? Make sure that it's free of moisture, because next step you're going to add something to digest it and to leach the metals. After you have dried the soil, right, you place into this oven for a week, a fan oven, okay, which means this oven is very specialized. It contains chemical. You don't want it to be the oven to dry your glassware, no interchange, no contamination. Then you grind this very hard now, but very dry soil using mortar and pestle. First here, mortar and pestle, grind. Okay, no large lumps should remain after grinding. If not, you cannot get to the center of these lumps here. Right. You should not, when there's lumps, it means the soil is still very wet. Now next is what we do. How do we get out the metals? We will weigh about 0.6 grams of soil into 3 centrifuge tubes, right? Those new blue color tubes here, the cap. Then we add 12 ml of the 18 mega ohm of water with a kind of resistance, okay, to the first tube. We ensure well mixing, to stir it, mix it. And then we filter the mixture using this Whatman grade 42 filter paper with pore size, right, that has been washed with this very same ultra pure water into the 25 ml volumetric flask. Because concentration matters a lot, so it's important to use glassware that's calibrated like a VF volumetric flask. Let me mark up the water level, right, how you did your analytical experiment, and ensure that the mixture is mixed well. Okay, so you can watch the detailed clip over here. Hope, as I explained to you, you can get a picture, you can jot down, link it back to your prior experiences and knowledge, what you have seen in the lab. Now for the second tube, you add an amount of the multi-element standards. So there's always standards given to you because you are trying to compare against the sample. Perform the same steps as the previous slide, okay? Then for the third tube, you add 8 ml of aqua regia and then you gently mix to ensure that the soil are wetted properly. Make sure every part of the soil has a bit of wetness from the concentrated acid. Aqua regia, mixture of corn nitric acid, corn hydrochloric acid, 3:1. Then you proceed with this milestone. You see, you wet it, right? Put inside, right here. Open the cap, put inside there, inside. This is where it's sample. It's being scanned. There's a protocol. Okay, check out another video, but it's not tested in this module. It's for lab, but I want to give you a bit of coverage. Use this plastic cap on the centrifuge tube when you transport the mixture. Then you place the third tube in the soil digester. Okay, digester. After that, decant the sample from a tube to another tube. Okay, filter using a syringe and membrane. Right, these are things you can try in the lab. Next time, go to the advanced analytical lab. Please ask the lab technicians, have a look. Okay. Do the same thing, right? And then always prepare blanks, but what are the blanks you want to use is dependent on the research question. Okay. Now, let's move on to the instrumental analysis. The instrument, we use ICP-AES. That's how it looks like. A brand we bought in the same lab. Now we have the 4 sample solutions that I prepare. Okay. Then you place them inside there. And then you have auto sampler that can accommodate up to 60 samples per run. Overnight you can do that, go for lunch and come back. Don't waste time. Have a look at the trailer, how it looks like. So next time you're a tester, you can say, oh, what instrument is this? You don't say it's a GCMS. Oh, this is a ICP-AES. What is it used for? Soil sampling. You have standards for heavy metals to check about soil. Long clip about how to use it is over here. Feel free again for interest. So now it's your turn to do your work here. Get your brain working hard and get your hands dirty. You come up with your own research questions. Okay, one question and bring to the class. And if asked, be ready to share and discuss. What are you curious about? Why you want to determine the concentration of certain elements in the soil? What do you want to investigate? What data will you need? Right? So there's a potential activity that will give you a sample of lab data from previous cohort, analyze it, and hopefully that can answer your research questions. And with that, this is my last slide. I close it with the possible sample solutions that you might get here. Different tubes, right? Different concentration of the blank and the content it has inside here. Standards, the different concentrations. What will you do? Okay, I already give you a bit of preview of the purpose. But then the next step is to evaluate, right? To link it to how do you answer your research question that's of most pertinence to you. Okay, so I'll see you in class then. Cheers. Bye-bye.